Good morning, students. In this class, we are going to discuss about linkage and various types of linkage. So, definition of linkage I have already explained in the previous class. So, let us see once again what is linkage. Linkage may be defined as the tendency of linked genes stay together and transmit generation to generation and transmit generation to generation is called linkage. Now let us see there are two types of linkage: complete linkage and incomplete linkage. So linkage are two types. First one is known as the complete linkage and other one is known as the incomplete linkage. So let us see types of linkage. The first one, complete linkage. Okay, complete linkage. So first of all, you have to know definition. The linkage in which the genes are closely related to each other, present on the same chromosome, and there is no crossing over, and only parental combinations are possible, known as complete linkage. Once again, what is complete linkage? The linkage in which genes are closely located on the chromosome as a result only parental combination is possible. Parental combination is possible in absence of crossing over. In absence of crossing over is called complete linkage. So once again, what is complete linkage? The linkage in which genes are closely located on the chromosome. As a result, only parental combination is possible. In absence of crossing over is called complete linkage. Let us see by taking example of complete linkage. We will take a pair of homologous chromosomes on which, you will take a pair of chromosomes on which genes are closely located. Suppose that there are two chromosomes. This is centromere, this is the chromosome. This is the chromosome number first and this is the chromosome number second. So first of all, the DNA content become double. That means, and there are two genes. Suppose that there is a gene A and there is gene B that are closely located to each other. Gene A and gene B. Gene A and gene B. And here the homolog is present. Gene small a and there is small b. So first of all, there is replication takes place. Replication. Or you can say duplication takes place. The DNA content become double. DNA content become double in the cell. So this is also known as tetrad formation. Okay, so there will be capital A, there will be again capital A. Okay, and there will be capital B, and there is capital B. In this case also, the DNA content become double. Okay, so there will be small a and there is small a, and here is small b and there is small b. Genes are closely located on the same chromosome. Such type of genes known as linked genes. Already explained the definition. Now let us see the next diagram. We will continue downside. So this is the step 1 and this is the step 2. Okay, so in the step 1, this is the pair of homologous chromosome and these are linked genes. Why linked genes? Because they are closely located on the same chromosome, so it is known as linked genes. And such pair of chromosome known as homologous chromosome. Let us see, we will continue, move down. The third diagram. In the third diagram, okay, genes are closely associated. As a result, there is no crossing over. Genes are closely related to each other, so as a result, there is no crossing over. Okay, in the third diagram, genes are closely located to each other, so there is no crossing over. Okay, we have already drawn in the step 2. Okay, so there is the genes is capital A and there is capital A and there is small a and there is small b. Sorry, there is capital A, there is capital A, there is capital B and there is also capital B. In this case, small a, and there is small b and small b. In absence of crossing over, because the genes are closely located, so there is no crossing over between the two non-sister chromatids. As a result, the gametes produced by this organism containing chromosome, that is only parental combinations possible. It will split lengthwise and it will produce two types of gametes, that is having gene capital A and capital B. Here will be also, in absence of crossing over, only parental combinations are possible. That means the genes that are present on parents is also occurs in the offsprings after gamete formation. In this case also, when it will separate for gamete formation, so it will produce also parental combinations. Okay, so it will be capital A, small b. Here also capital A, small a and small b. So once again, first of all, a pair of chromosomes. This chromosome will double by DNA replication. Then in absence of crossing over, why crossing over is absent? Because genes are closely located. As a result, there is no crossing over. So after gamete formation, you can see all are the parental combinations. P stands for parental. This is the parental because the same combination contains the parent. This is also parental. This is also parental combination, and this is also parental combination. So once again, we call it equation the linkage in which the genes are closely located to each other. As a result, only parental combinations are possible. In absence of crossing over is known as complete linkage. It is very less common in nature, that is found in Drosophila. Let us see second type, that is incomplete linkage. Okay, incomplete linkage. So first of all, you have to write definition of incomplete linkage. The second type of linkage, incomplete linkage. Incomplete linkage, the linkage in which crossing over possible between the pair of chromosome, because the genes are distantly located between the pair of chromosome. As a result, both parental as well as new combinations. That means recombinants, new combinations are possible, called incomplete linkage. Once again, what is incomplete linkage? The linkage in which crossing over possible takes place between the pair of chromosome. Why? Because genes are distantly located. Okay. As a result, both parental as well as new combinations, that is recombinants, are possible and produced, called incomplete linkage. You can see by taking example. Okay. Let us see incomplete linkage example. Again, we will take a pair of chromosome having gene capital A, capital B, and small b. Let us see. Again, we will start from the same steps that we have done in the complete linkage. This is a pair of chromosome. This is a pair of chromosome where genes are distantly located. There is certain distance, capital A and capital B. Okay, and there is capital A and capital B. So you can see again in the diagram also. Okay, genes are distantly located to each other. Okay, let us see. Genes are distantly means there is a distance will be takes place between the genes. Capital A, capital B. Here small a and small b. Sorry, small a and small b. In the previous one, genes are closely located. As a result, there is no crossover. But in this case, you can see the genes are apart. There is a certain distance between the genes that is responsible for crossing over between the chromosomes, between the genes. So let us see. First of all, the DNA content become double. So here you can say DNA replication will take place, or you can say replication. So after replication, you know DNA content become double. So it will become double like this. This is the centromere. Okay. So there will be capital A. There is capital B. There is also capital A and there will be capital B. In this case, the same thing will be takes place. Okay. After doubling, DNA content become double. So there will be small a. Okay. And there is small a and there is small b and there is small b. So step 1 is the DNA replication and formation of tetrads. In the next step, there is exchange of genes takes place between the non-sister chromatids. You know this pair is known as sister chromatids and this pair also known as sister chromatid. but these two pairs is known as non-sister chromatids. That means the chromatids of different pair is known as non-sister chromatids. In the step third, there is an exchange of genes takes place between the non-sister chromatids. So let us see in the step third. In the step 1, DNA replication takes place. In the step 2, okay, in the step 1 DNA replication takes place. In the step 2, there is a crossing over. So let us see next step. The crossing over takes place between the non-sister chromatids. So again we will draw the same diagram, but you will show the crossing over between the non-sister chromatids. Okay? Again we will draw the diagram, but you will show the crossing over. Okay? So there is crossing over takes place between these chromatids. Crossing over will take place between these chromatids. Okay? So there is gene will be there, capital A, there is capital B. There is also capital A, there is capital B. In this case, small a, and there is small b, and there is small b. After crossing over, you know, crossing over takes place between the two non-sister chromatids. So it will produce two types of gametes. Okay? So when it will splits, then it will produce two types of gametes. The first one is capital A and capital B. This is the parent combination because it present in the parent. But second one is small a, capital B. Second one will be small a and capital B, that is recombinant. So let us see second one, small a and capital B. Some part of capital A, small b. Some part of small b transferred in this case here. Capital A, small b. So here you will write capital A and small b. So this is recombinant because new combination produced in the earlier there is capital A, capital B. According to complete recombinant there should be capital A, capital B, but they should exchange again capital A, small b. Now let us see third combination. When this chromosome will splits, it will produce small a, capital B. Okay, small a, capital B. Third combination, that is small a and capital B. Small a, capital B. This part exchange of genes takes place. So there will be small a and there will be capital B. Small a and there is capital B. And last combination will be small a, small b. Last combination will be small a and small b. Small a and small b. So you can see in the previous one, in absence of crossing over, only parental combinations are possible. But in this case, due to crossing over takes place between these two non-sister chromatids, new combinations are possible. Out of 4, it is parental because it is similar to the parent. But this is recombinant. New combinations possible. In this case also, new combinations possible. So it is also recombinant because capital A is small b. This is new combination. In this case also, small a, capital B. This is also a new combination. And our fourth one, it is also parental. So again, we will recall the definition: the linkage in which crossing over takes place between the two non-sister chromatids as a result, parental as well as new combinations are possible, known as incomplete linkage. Okay, it is more common in the nature. So this is all about complete and incomplete linkage. Let us see the difference between the complete and incomplete linkage. Let us see the difference between complete and incomplete linkage. So you can differentiate by making two columns. One column for complete linkage and other column for incomplete linkage. Okay, complete linkage. Complete linkage. And here you can write incomplete linkage. Incomplete linkage. Okay. So incomplete linkage, you know, in case of complete linkage, the genes are closely located. Genes are closely located on the chromosome. This is the first difference on the basis of distance between the linkage. In case of complete linkage, the genes are closely located on the chromosome. In this case, the genes are distantly, means distance is there, distantly located on the chromosome. This is the first difference. Second difference, in which case crossing over takes place? In incomplete linkage, crossing over takes place because distance is more. When more distance, crossing over will take place. So no crossing over takes place. No crossing over occurs. No crossing over occurs. But in this case, crossing over takes place. Crossing over occurs. Okay? Third difference. You know, in complete linkage, in absence of crossing over, in the complete linkage, only parental combinations are possible. So only parental combinations are possible. In which case, in complete linkage. And in this case, both parental as well as new combinations are possible. Both parental as well as new combinations are possible. New combinations are possible. Okay? And fourth and last difference, complete linkage is very less common. It is less common. Like in case of Drosophila, it is less common. It is more common. It is in complete linkage, it is more common. So this is all about linkage and various types of linkage. Once again, you can differentiate on the basis of complete and incomplete linkage. First one, distance. Genes are closely located on the chromosome. In case of complete linkage. In case of incomplete linkage, genes are distantly located on the chromosome. That means there is a certain distance between the genes that is responsible for crossing over. Second one, no crossing over takes place. Here crossing over occurs. Third one, only parental combinations are possible. Why parental? Because in absence of crossing over, only parental combinations are possible. But in this case, both parental as well as new combinations are possible. And last one, complete linkage is very less common. Like in case of Drosophila, we will discuss about during the Marvel's experiments. But in this case, it is more common. So this is all about various types of linkage. Thank you.